minor in health sciences. Over the past couple of years, I have had the great privilege of working with my mentor and PI, Dr. Rakesh Tiwari, over at Chapman University School of Pharmacy. And today I am thrilled to be presenting the culmination of my work for the past few years on the tumor-targeted delivery of siRNA via fatty acyl conjugates of cell-penetrating peptides. Here's a little roadmap of my presentation. I will first begin by explaining the background and rationale of our study. Then we will dive into the results and data analysis, and end with a few key takeaways and forthcoming plans for our lab. To begin with a little introduction, why is our lab interested in siRNA therapeutics for the treatment of cancer? As we all know, chemotherapy comes with a multitude of side effects, yet it is still the mainstay approach to treat cancer today. With the dire need of a more novel treatment method, siRNA therapeutics are an emerging option for multiple diseases, one of which is cancer. And siRNA therapeutics will act by inhibiting overly expressed cancer-causing proteins at the source, which is the mRNA. And this is unique because most traditional drugs will actually target the proteins. Now going into the mechanism of action, I really want to highlight this first point here, that siRNA therapeutics follow a naturally occurring phenomenon that effectively silences genes in mammals. So normally, the silencing is happening through a mechanism which is presented on the left side of this figure here, in which the cell prepares microRNA that incorporates into a protein complex called RNA-induced silencing complex, RISC for short. Following a few steps, part of the microRNA is lost to form the mature RISC, and it will then go and find the mRNA that I was just telling you about to degrade it and effectively silence the gene. And so in comparison, the mechanism for our proposed treatment utilizes synthetically produced double-stranded siRNA, and once delivered intracellularly, it will follow the same exact mechanism of incorporating with RISC, lose one of the strands to perform mature RISC in order to find the target mRNA and effectively silence the gene. And despite much promise in its ability to effectively silence genes, the delivery of siRNA to target cell remains a major obstacle for this type of treatment. You can imagine that when you introduce any type of drug into your body, there will be various obstacles. And so here, if I could direct your attention to the figure on the right, some extracellular barriers include siRNA degradation through endogenous endonucleases, uptake and clearance through the reticuloendothelial system, as well as charge repulsion of siRNA with the cell membrane. If taken up, there are some intracellular barriers as well, such as endosomal entrapment, which could lead to exocytosis or lysosomal degradation. Despite its emergence as a new type of therapy, siRNA faces many obstacles in its delivery. Thus, there is a dire need for an efficient delivery vehicle. So one way to overcome these barriers is use of carriers and transfection agents. These carriers are thought to protect the siRNA from the extracellular barriers that I was just mentioning. And once uptaken into the cell, they have a mechanism in which they can efficiently release the siRNA. And our lab specifically looks at cell-penetrating peptides as a delivery agent due to its characteristics of being less toxic, enhanced cellular uptake, and easily synthesized in the laboratory. So this study aims to investigate the binding and delivery of siRNA to tumor cells using fatty acyl RN HR4 peptide conjugates, where N is equal to 0, 1, 2, 3, 4, or 5. We hypothesized that cell-penetrating peptides containing positively charged histidine and arginine amino acids conjugated with an oleic acid will increase the peptide conjugate's amphipathic nature for efficient binding with siRNA, cell membrane penetration, and lower toxicity. So here are a few examples of the peptide structures in the corresponding MALDI mass spectrometry results. So we have C18HR4 here. We see the alternating arginine and histidine amino acids with the conjugated C18 oleic acid. And so how we verify that we were successfully able to produce these peptide conjugates in our lab is through MALDI mass spec. And so we see the exact mass here of the peptide 1455. And what this machine tells us is whatever we produced in the lab, we put it on a plate, and then the machine will tell us the exact mass. One thing to note here is that the MALDI mass spectrometer will tell us the mass of the peptide plus 1 hydrogen, So that is the reason you see 1456 on the MALDI mass spec in comparison to the 1455. Similar results can be seen for our second peptide, C18R3HR4, which has 3 extra arginine residues. We see an exact mass of 1923, but in the MALDI mass spec, it will show us 1924. But these are verifying that we were successfully able to produce these 2 peptides in our lab. As mentioned before, though, due to time, I only limited to 2 peptides, but we were successfully able to produce R1, R2, R4, and R5 C18HR4 peptides as well. Following the production of our peptide conjugates, we moved on to a cytotoxicity MTS assay. And so for this assay, we used MDA-MB231 cells, which are triple negative breast cancer cells, and they are known to metastasize very quickly in one of the most aggressive forms of breast cancer. And so in this assay, we see the y-axis is % cell viability, and the x-axis has the different treatment groups. So the negative control non-treatment group was used as a reference point where all these groups were 100% cell viability. The positive control for cytotoxicity was 30% DMSO solution, and we had different N to P ratios of our cell penetrating peptide conjugates. So the N in N to P ratio stands for the ionizable amine of our peptide in the amino acid portion. And then the P represents the ionizable phosphate group in the siRNA. And so one thing to note here is that the siRNA concentrations were kept constant throughout all of these N to P ratios, but the peptide concentration was increased in order to increase these N to P ratios. Now, looking at the overall graph, we see a trend here where with increasing N to P ratios, we see that the peptides start to become more and more toxic to the cells. So we made the cutoff point at around N to P ratio 40, where we see about 80 to 85% cell viability. And anything above this N to P ratio was considered to be too toxic for clinical use. We followed this up with a cellular uptake FACS flow cytometry assay. And so this is to ensure that the peptide conjugates are efficiently delivering the siRNA into the cells. We used a Alexa 488 fluorescent tag, and we see the mean fluorescence on the y-axis along with all the different treatment groups on the x-axis. The CD20-CGKRK is a known peptide to efficiently deliver siRNA into cells. So we used it as a positive control, and we had the 2 negative control groups of no treatment and just the Alexa 488 siRNA itself. And so we see another trend here with the increasing arginine residues at the end of HR4. We see better and better cellular uptake. And with the C18R4 HR4 peptide, we see greater than double the amount of Alexa 488 tagged siRNA delivered into the cells. And this is verifying that, again, our peptide conjugates were successfully able to deliver the siRNA into the cells. One drawback to this assay is that we can't tell where specifically in the cell that the siRNA was delivered to. So this will need to be followed up with a confocal microscopy where we can see the exact location of the siRNA. Following this, we performed a binding affinity assay, a gel shift assay, where we see the N to P ratios labeled above each well. And we can tell that there is no peptide in the N to P ratio 0 due to the beautiful siRNA band shown here. This is used as kind of a marker for the rest of the peptide conjugate siRNA complexes. And so we see another beautiful trend here that with increasing N to P ratios, meaning more peptide in each well, we start to see the siRNA band diminish. And this means that the siRNA band has shifted due to binding with the peptide conjugate. And so an average of about N to P ratio 2 can be seen where the siRNA bands diminish for all these different peptide conjugates. And so we concluded that when N to P ratios 0 to 40 were tested, all peptides showed significant binding to siRNA at N to P ratios greater than 2. We further followed this up with a serum stability assay where we treated the cells with fetal bovine serum, a 25% concentration of FBS. And to put this in perspective, normal physiological conditions is about 10%, so we used much harsher conditions to see if the peptide conjugate was able to protect the siRNA for 24 hours. And following the 24-hour treatment, we treat the peptide conjugate and siRNA complex with heparin sulfate, which releases the siRNA from the conjugate. And so this is testing whether, this is representative of whether siRNA was released or if siRNA was degraded along with the peptide conjugates over these 24 hours. And these HBSS is a buffer, so it is a positive control to show what the siRNA band should look like. And then we again see a trend where increasing N-to-P ratios successfully protect the siRNA for a 24-hour period. And as an average, we concluded that anything greater than N-to-P ratios of 20 were able to successfully protect the siRNA. So a few key takeaways from this presentation. The C18RXHR4 fatty acyl cell-penetrating peptides showed significant enhancement of cellular uptake when increasing the number of arginine residues. All peptides showed significant binding affinity to siRNA at N-to-P ratios greater than 2. And following 24-hour treatment with FBS, gel shift assays revealed all peptide conjugates protect the siRNA at N-to-P ratios greater than 20. Some forthcoming plans is to evaluate the actual gene silencing of the siRNA of signal transducer and activator of transcription 3, STAT3 protein, SRC, and kinesin spindle protein using peptide siRNA complexes through Western blot. These proteins are known to be upregulated and overexpressed in various different cancers, so we want to be able to see actual gene silencing through this mechanism. We will further characterize the peptide siRNA complexes using zeta sizer and transmission electron microscopy. What I mentioned earlier, confocal microscopy to corroborate the data obtained through the FACS flow cytometry assay. And fingers crossed, if everything goes well, in vivo studies in mouse breast cancer models. I wanted to take a moment to thank the Schmid College of Science and Technology for the continued support through my undergraduate career, Chapman University School of Pharmacy for providing me the opportunity to do this research, as well as Center of Undergraduate Excellence, my lab mentor, Mohammed Imran Sajid, and principal investigator, Dr. Rakesh Tiwari. Thank you for listening, and I would be happy to answer any questions.
Shun, thank you very much. We would be very happy to take questions from folks in the audience. If you want to just unmute yourself, or if you prefer, I can read something out of the chat. Shun, perhaps I could lead off on the questions. Could you tell us a little bit about fatty acyls and why that's an appropriate candidate compound to use for this approach as compared to other potential compounds, or tell us more about other potential compounds that you could imagine working for this?
Yes, definitely. So the reason we used fatty acyl compounds is, so the reason for the positively charged amino acids is to bind with the negatively charged siRNA. And then we believe that this long chain of fatty acids will actually wrap around to kind of form a covering on the siRNA to protect it from the extracellular barriers. And we also believe that the hydrophobic nature of the oleic acid will help in the cell penetration of the cell membrane. So we chose to use a fatty acyl group along with the hydrophilic amino acids or positively charged amino acids to get the synergistic effect of cell penetration and binding to the siRNA.
Have there been other candidates that have been identified by other groups?
Yeah, there are a lot of other groups researching cell penetrating peptides right now. We also see the use of tryptophan replacing some of the amino acids, which could also work with the binding to the siRNA. And a lot of groups do use histidine though, because one of the barriers that I mentioned, the endosomal escape, it is thought that histidine, due to the difference in the extracellular and intracellular environment, will have some sort of effect in releasing the siRNA from the endosomes, although the mechanism is not fully understood.
We have time for another question if anyone has one from the crowd. You have received many congratulations already in the chat, but please join me in a virtual round of applause, Shun, for everything that you've achieved in the last 4 years. My congratulations and best wishes for everything that's to come.
Thank you so much, Dr. Goldsmith, and thank you everybody for listening to my presentation.
We are sticking in the pharmaceutical space for another presentation. This this morning session has some great variety of different presentations, and so we're next going to hear from Johnny Atwal, who's going to talk to us about pharmaceutical compounding in veterinary medicine. He's finishing his bachelor's in biochemistry and molecular biology as well, also minoring in health science. And Johnny, the floor is yours.
Great, let me get my slides up, and I would appreciate if you can confirm that you can see them. Okay.
Perfect.
Okay, great.
Everyone can see gabapentin on the screen.
Perfect, perfect. Hi everyone. So my name is Johnny, and wait, let me just make sure. Okay, yes. So hi everyone, I'm Johnny, and I'm going to be talking about compounding in veterinary medicine, but also taking a focus on gabapentin. And so to start off, I'll talk a little bit about the project goals we had. So I wanted to learn more about veterinary compounding and its needs and limitations. This is an extremely commonly occurring practice in veterinary medicine, yet it is very, very understudied. And so I was eager to dive into the literature and learn as much as I could about it. Gabapentin then fits really well into this project because on the veterinary side of things, it exists almost exclusively in its compounded form. Animals are very rarely prescribed a gabapentin pill or tablet the same way that it would be given to a human. And so the compounding process is frequently used with this drug so that animals can take it. And then the third goal was to get in the lab and to use the HPLC to analyze the potency of various gabapentin formulations. I was able to get in the lab, but unfortunately, due to some COVID restrictions and stuff, I wasn't able to get enough data to present during this presentation. But not to worry, we'll discuss many other important things. So integral to the understanding of this project is what is compounding. And so compounding is essentially any modification that is made to a drug. And this can be as simple as adding a flavor additive to entice the animal to take the drug, or it can be as complex as combining numerous therapeutic agents together and creating a new drug through that, if you will. And so the next thing to keep in mind throughout this presentation are the US Pharmacopoeia guidelines. These are basically the only regulatory guidelines on compounded formulations, and they dictate that a compounded formulation has to have between 90 to 110% of the labeled dosage. And there's an easy example there. If the labeled is 50 milligrams per milliliter, the drug would have to have, the compounded formulation would have to contain 45 to 55 milligrams per milliliter of that drug. Any less, and you would risk a subtherapeutic agent that's not going to do what you would like it to do. And anything over that range, and you would risk toxicity or overdosing your patient, which is also not good and not wanted. And so a little bit more about gabapentin. In humans, gabapentin was first used as an anticonvulsant agent, but and then it was later found to be very effective in treating postherpetic neuralgia. Postherpetic neuralgia is a complication of shingles in adults, and it's excruciating nerve pain that is chronic. And so it was demonstrated in humans to also be very efficacious in managing chronic pain. In felines, it's the most commonly prescribed medication for musculoskeletal pain. And the FDA has approved tablets, capsules, and oral solutions, but of course, these are approved for use in man. Pharmacists and veterinarians then take these tablets, and either the tablets or the capsules, both can be used, and are able to then make oral suspensions of gabapentin so that our animal patients can take this drug. And so to talk a little bit about the pharmacokinetics and dosing, in the most recent study on the pharmacokinetics of gabapentin in cats, they actually found that there was no bioavailability via the transdermal route. They only found some bioavailability via the oral and intravenous administration. But it's important to note that the transdermal agent they used was also compounded. And so there's no real way to know that if this is because of the transdermal drug delivery, or if it's because of the actual compounded formulation, since in humans, there is some work delivering gabapentin transdermally. And so the current dosing practices in felines and canines are 10 milligrams per kilogram of body weight twice daily orally. This can vary patient to patient, but it's pretty consistently around this dose. But the EC50, which is the half maximal effective concentration, this number just gives us an idea of what dosage will lead to the beneficial effects of the drug. This has not been determined in felines and canines. If you can see from the bar chart, we have this data for humans and rats, and we can then anticipate that the values for canines and felines will fall somewhere in between. However, the research to quantify these values has not been done. And so what that tells us is the dosing practices in small animals, which is based on the human and rat model, might not be accurate. And so there are some scientists that are hypothesizing that there might be, it might be necessary to alter the dose in order to have the drug be more effective in managing pain. This would be the new proposed dose, but of course, this would need to be tested in actual a set of canine patients, feline patients, and to quantify that data in those species. And what we can see in the bottom bar chart is the pharmacokinetic differences of processing this drug in cats and dogs. What we see, so the Cmax is the maximal concentration of the drug in the blood. And what we see is these are different values for the same oral dose, and they occur at different times, which is what the Tmax is. And all of this affirms is that these different species are processing this drug differently. And so it begs the question, why are we using the human model and the rat model to determine dosing for felines and canines when we have sufficient data to suggest that all of these species are processing this drug very differently? And so to talk a little bit about neuropathic pain, which is what gabapentin seeks to resolve, there's an occurrence of a spinal cord injury, and then there's an upregulation of these calcium alpha 2S1 subunits. I'll be discussing those more deeply soon, but the upregulation of these subunits, these proteins, is what leads to neuropathic pain, which can present either as tactile allodynia, which is a painful response in the patient to a non-painful stimulus, or hyperalgesia, which is an exaggerated pain response to a minimally painful stimulus. When the onset of these occurs, we seek to treat it with pharmaceutical agents, commonly gabapentin, and this leads to the reversal of those neuropathic states. But this is where it gets a little hazy, because the mechanism of action for gabapentin, despite how well researched it is not fully known. And so this is just some data from the literature that supports what I presented in the last slide, how there is the upregulation of those calcium alpha 2S1 subunits. And in this study, they had rats, and they gave these rats a spinal cord injury, which led to allodynia. The sham rats are the rats in the control group, so they don't have a spinal cord injury. And then allodynic rats are the ones that do have the spinal cord injury, and on top of that, have neuropathic pain, whereas non-allodynic do not have neuropathic pain. And we see that with the pain and the spinal cord injury, there is the upregulation of those proteins. And then in this graphic, all we can see, this is a pain withdrawal test, which it just provides the rat with a stimulus. And once the stimulus becomes too painful, they can remove their paw. And what we see is with the administration of gabapentin, so post-injection after gabapentin is able to take its effect, they are able to withstand higher pain stimuli. And so this brings us to kind of the most plausible mechanism of action, that the calcium ion will bind to the alpha 2S1 subunit before it's trafficked through the channel. And there is an upregulation of the production of these alpha 2S1 subunits when a spinal cord injury has occurred. And then this leads to an increase of those calcium currents. When gabapentin is taken, it can bind to the alpha 2S1 subunit and then block the channel. This has been validated in studies that show patients taking gabapentin have a reduction of those calcium currents, which we know can be measured since when calcium moves across a membrane, we see there's a measurable difference in the electric potential of that membrane. And so moving on to, for me, the really, really exciting part of this project, since, next year I'm going into veterinary medicine, is, why compounding is so important and why it's here to stay, despite some of its issues. And this basically boils down to two main factors. Veterinarians seek to treat an extremely large number of species that are afflicted with an even greater number of maladies and illnesses. And so we really need a rich variety of pharmaceutical agents to do this successfully. And we find that rich variety in the approved human medications. Now, on top of that, there's a lack of financial incentive for pharmaceutical agencies. There's a lot more people buying drugs for other people than people buying drugs for their animals. And so the profit margins with producing a veterinary drug are much reduced, which reduces the incentive to produce them. On top of that, even if a veterinary pharmaceutical company was to pursue the approval of a drug, the chances that the human generic compounded version would be cheaper are fairly high. So it leaves compounding as an economically feasible option for a lot of pet owners. And so instead of doing away with the practice, we really need to work to standardize it and make sure that we are doing this process very efficaciously. Because unfortunately, I'll present on one, but there have been numerous cases recently on malpractice in compounding in veterinary medicine. The Burton et al. 2016 case, this was a group of vets in UC Davis, and they were treating a bunch of cancer patient dogs with the oral chemotherapy drug lomustine. There was a shortage in the market, and so they resorted to using the compounded version as opposed to the FDA-approved version. And so what they found was the dogs taking the compounded version had reduced neutropenia. Neutropenia is a side effect of taking this chemotherapy agent, and it's a reduction of the neutrophil count in the blood. And so it's a pretty serious side effect, but it's very commonly observed when taking this drug, and veterinarians have gotten really good at managing this side effect. And so even though the reduction of these side effects might seem positive, it actually indicated to the veterinarians that the drug is not in high enough concentrations to elicit the anti-cancer effect. And so to prove this hypothesis of theirs, they acquired samples from 5 compounding pharmacies and were then able to test them using HPLC to test the potency. Unfortunately, they found that only 1 pharmacy out of the 5 was able to provide a number of samples whose potency was within the appropriate dose range. So this is really concerning because it proves that the patients receiving these compounded drugs were either receiving too much, which could be deadly, or too little, which would mean that their cancer is not being combated effectively. And so to quickly go over the methodology that Burton et al. And myself and many other studies have adapted to test this sort of thing is you would acquire the standard of the drug, as I did from Sigma Aldrich. You would make a series of dilutions of known concentrations and then put those through the HPLC, which would allow us to get the area under the curve generated from the peak of that HPLC chromatogram. And then we can make a standard curve which relates the area under the curve of the HPLC to the known concentration. From that calibration curve, we can then get our standard equation. And then when we run the unknown samples through the HPLC and get the area under the curve from that data, we can use the equation and calculate for our concentration. Then when we have our concentration, we're going to refer back to our US Pharmacopoeia guidelines and see if it was compounded effectively. And so just to wrap up with some future directions, there needs to be a lot more research into the potency of compounded drugs. There needs to be frequent and random testing of drugs coming out of compounding pharmacies to ensure that they're within the appropriate ranges. On top of that, there needs to be a lot more oversight and regulation. This is such a common practice in veterinary medicine, yet there's virtually no oversight or regulation. And on top of that, there needs to be species-specific research. The use of the human and the rat model to inform dosing practices in veterinary medicine is outdated, and it's doing our patients a disservice since we have no real data to support that the dosing practices that we are using currently are the optimal, most effective doses to lead to pain relief in the example of gabapentin. So thank you so much, and I'm happy to take your questions now.
Excellent. Thank you, Johnny. And we'd be happy to take questions from the crowd. You can unmute or feel free to type something in the chat. Mr. Winston.
Fabulous presentation. It was particularly interesting because gabapentin is also being prescribed by veterinarians as a mild sedative for dogs, as well as being used for pain management. I found it particularly interesting that when you were looking at your time-based effectiveness for pain control, that actually maps virtually identical to the effective time that the sedative is effective as well.
Yeah, that is really interesting. Yes, thank you for that comment.
Yeah, it's about 3 hours, 3.5 hours or so, and you notice that it wears off.
Johnny, do you think that these differences in efficacy, this interaction between time and efficacy, is a function of the metabolic differences at the scale of these calcium channels? Or so that would be a very molecular level, you know.
Right. Yeah, I understand.
Effect.
Right. I understand your question. In all honesty, it was outside the scope of this project, and I'm not 100% sure. There basically is just so little data on like species specific. So there's no data that quantifies the effective or the concentration of gabapentin in canine and feline blood that would lead to the optimal level of pain relief. And this is the case for numerous drugs across the board in veterinary medicine. We just don't have the information. The research hasn't been done to just learn how these drugs are interacting in animals. We have a lot of data with rats, and we have a lot of data with humans. And for a long time, veterinarians have just hoped that we can extrapolate that data to make it work. And obviously, we wouldn't be prescribing these drugs if we weren't seeing the positive benefits. But there's no way of knowing that we're at the optimal dosing, the most optimal dosing.
Other questions? Dr. Keller, I see you popping up on the screen.
Johnny, great presentation. Can I follow up on that last comment that you made and last question from Dr. Goldsmith? So this species-specific thing seems really challenging to me. Does this mean that you have to go through and do this for every species that a veterinarian might treat and every medication that a veterinarian might use? Or do you think there's a chance that there are broader patterns, that once we do this with a subset, we can start to pull out what, you know, ratios on body to volume, mass to volume looks like? Are there ways that you can do this that doesn't require, you know, a complete test for every single species and every single drug?
Right. I would imagine there is. I would not be able to tell you specifically, you know, the groupings of drugs that would work, but I'm sure that this is the case, that, you know, drugs exist in groups, you know, based on similarity of their structure. You know, for example, off the top of my head, you know, I would imagine that you would not need to test, you know, every single ACE inhibitor in veterinary medicine. You would be able to test one ACE inhibitor, how it interacts with feline and canine metabolism, and then use that data to make, you know, to apply it to all ACE inhibitors. Just a quick example off the top of my head. I imagine that would be the case, and yes, it would not be feasible to conduct a research study for every single drug with every single species.
Great answer. Great presentation. Well done.
Thank you.
I hope everyone will join me in congratulating Johnny on a great presentation. And as with Shun, we wish you all the very best, and our congratulations on all of your time at Chapman. And we'll look forward to hearing about your adventures in veterinary medicine very, very soon.
Thank you. Thank you.
With that, we are right on time for our next speaker, who is going to talk to us about investigating potential biomarkers for methotrexate-induced neurotoxicity. As you can see all of our graduates today are working on very simple concepts that do not involve any hard words that I'm forced to pronounce on the spot. So far, I think I've done okay, but we'll see. So Edena Khoshaba is finishing her bachelor's in biochemistry and molecular biology, and a minor in rhetoric and writing and leadership studies. And it's really a pleasure to see her presentation. And Edena, the floor is yours. We'll see if we can unfreeze Edena here and unmute. Uh-oh. We've got technical difficulties, I believe. Let's see if we can get Edena back on the screen. And while we do that, let's see, I see her coming back. You can all have a brief moment to look at me doing my undergraduate research in Barrow, Colorado Island in Panama. And I gave my honors thesis presentation 16 years ago to the day. Let's see if we've got Edena back. No, Edena, we can't. Can you try saying something out loud for us? Or that looks promising. Or I thought I saw that maybe you've got Kristen signed in on as well. Cailyn, are you in-house? Do you want to switch? I'm a little bit reluctant to do that if you've got friends or family that are planning on attending, but I do see Dr. Atamian at least is here.
I can. Can I get like a couple seconds just really quickly to make sure someone has the link?
Sure.
Okay, cool.
We'll
Thank
see
you.
if we can get Edena and the first person to the punch can go. It really wouldn't be 2021 without at least a technical difficulty. We'll get this out of the way as quickly as we can.
Okay.
Part of the normal now.
Sorry, I joined Kristen's hotspot. So how is it working now?
We can hear you perfectly, and let's see if you can share screen.
Okay, perfect. Let's see, I'm so sorry about that. Our Wi-Fi just like is the worst at all times, so.
That is quite okay, and because we've got the break coming after Cailyn, we'll just make sure that you have all the time that you need to present and all the time that Cailyn needs, and we'll run a little bit into break, and that's no problem. Without further ado, Edena, the floor is yours.
So yeah, I'm Edena. I'm a senior biochem major. I did this project at Baylor College of Medicine my sophomore summer, so it's been a while. Due to COVID, I could not do like my other research. So this was on investigating potential biomarkers for methotrexate-related neurotoxicity in acute leukemia patients at Texas Children's Hospital. So I know that's a mouthful, so we're going to break it down in this presentation. So before getting into the project, I want to give a brief background on acute lymphoblastic leukemia. This is the most commonly diagnosed cancer in children and is a type of cancer that is characterized by the bone marrow creating too many immature lymphocytes that can either come from a B or T cell lineage. As the number of leukemia cells increases, we can all see how if there's less room for healthy cells, this can lead to a variety of infections and other complications. However, amazingly, due to new treatment, the cure rate has reached up to 90% for long-term survival. But although we have a very high long-term survival, the amount of late effects and just treatment complications that go into the future are high. So methotrexate is one of the important chemotherapeutic agents used in ALL. It is an anti-folate agent that is able to interfere with DNA synthesis, which can stop leukemia cells from reproducing. However, methotrexate is also linked to cases of neurotoxicity is defined as any neurological change within 14 days of administered treatment. This can be presented in a variety of ways, such as stroke-like symptoms, seizures, aphasia, and encephalopathy. These neurological changes can lead to treatment delays, which can jeopardize long-term survival and even increase the risk of relapse. There isn't much information on the mechanism of methotrexate neurotoxicity, so our study was designed to find biomarkers that could help predict neurological impairment in pediatric ALL patients in the cerebrospinal fluid. And the reason we chose the cerebrospinal fluid was for two reasons. So for one, it is a biologically relevant sample for studying nervous system toxicity. And on the other hand, it is also readily available because when children receive intrathecal doses of methotrexate, they also withdraw cerebrospinal fluid to kind of counteract that balance within their body. So that is why we chose the CSF as a potential sample. However, the group does have other samples, including urine, saliva, blood, that they're also doing these same analyses on to try to create a more complete picture of what is happening in the body of these pediatric cancer patients that are having these interesting cases of neurotoxicity. So in order to study this, we used a global metabolomic and proteomic approach. And you might be familiar with other types of omics, such as genomics, transcriptomics, or proteomics. Each field has its own benefits and drawbacks. Together representing this sort of continuum from DNA all the way to these small biochemicals in the body. These small biochemicals are also known as metabolites, and they are the byproducts of cellular metabolism. One advantage of metabolomics is that the abundance of a metabolite is directly representing the molecular phenotype. So it's kind of like a snapshot of what's happening in your body at that time, so it can explain these complex cases of neurotoxicity. At my summer at Baylor, I did the metabolomics, and this past semester, I had the proteomics data to kind of like work on this project a little bit more since it's been a while, and I wanted to be more familiar with the aspects since it's been like 2 years since I worked on this project. So for my project in particular, we wanted to do an untargeted screen of the CSF for all the metabolites and proteins in it at the time of abstraction. So these metabolites, like I said, can provide novel insight because they respond rapidly to physiologic changes. So they serve as biomarkers for complex phenotypes, and one example of that are these cases of neurotox. In many like metabolomics and proteomics studies, patients are divided into what is called the discovery and replication cohort to provide another level of validation to the results. So what that means is if you have two separate groups of case and control, you can do the same analyses, and if results replicate in one study and into the next, that is for small sample sizes, it's the way to counteract the small sample size to see if results are still replicating. So the discovery cohort consisted of 12 neurotox cases and 22 controls, and the CSF samples were collected 90 days before or after an event. Whereas the replication cohort had 14 cases and 29 controls, and the CSF samples were collected within 30 days prior to an event. The reason for this design was to do an untargeted screen of metabolites in the discovery cohort and see which metabolites met our significance threshold of 0.05. We then took a closer look at those metabolites that did have one less than 0.05, and we see if in the replication cohort, if these results were also below 0.05, and if the direction of effect, looking at the fold change, was also in the same direction. So this is the sort of pipeline we did for the metabolomics analysis, and a similar one was used for the proteomics analysis. The CSF samples were first sent to Metabolon, where they performed a separation techniques such as ultra high performance liquid chromatography or gas chromatography, accompanied with the detection techniques such as mass spec. The data was then sent to us for further analysis, and 314 biochemicals were identified in the discovery cohort and 396 metabolites in the replication cohort. So since this lab was totally out of my comfort zone, like I had no experience in bioinformatics methods, coding in R, so it was a great experience. My background had been more in wet lab, so this was a great experience to learn something new because I was able to have the raw data sent to us from Metabolon, and I had to clean, organize it, prepare the data for analysis, run regression models, add covariates to see what can be a confounding variable. So it was just a different kind of science that I wasn't used to, and I fell in love with like the epidemiology and just getting large data sets and seeing how these trends can affect our pediatric cancer patients at Texas Children's. So here we have a table of our population statistics for the discovery and replication cohorts. Overall, study participants were mostly male, Hispanic, and treated in high or very high risk protocols. Of these demographic factors, we see the ones bolded. BMI, age, and ethnicity are significantly different between cases and controls in at least one of our cohorts. So as a result of that, we made sure to account for these as confounding variables in our linear regression and logistic regression analysis. So here's a volcano plot of the discovery cohort. The x-axis, as we see, shows the log 2 of the full change, and the y-axis is the negative log transformation of the p-value. By doing this negative log transformation, we can visualize smaller p-values as larger numbers and therefore towards the top of the plot. So these 8 metabolites shown in red had a p-value less than 0.05 and were therefore significantly different between cases, neurotoxic cases, and controls. And this is why we chose to see if these results would replicate in our replication cohort. So these are the 8 metabolites associated with neurotoxicity in the volcano plot. Of these 8 metabolites, we see malate here at the bottom that actually replicated. As we can see, the full change is 1.12, meaning it was a more highly upregulated in cases rather than controls. And we see that the p-value is also less than 0.05 in both cases. So I did a literature analysis on malate to see the possibilities of this being in our, like why this would affect neurotoxicity, and I'll get to that in a second. But I also wanted to quickly mention 5-methyl tetrahydrofolate, because even though this metabolite was not detected in a replication cohort, this is a very interesting metabolite to us because it's directly involved in the methotrexate action pathway, and it is reduced significantly when the drug is administered. So I think it is interesting that the full change was less than the one in our discovery cohort because it indicates that it was less abundant in cases and controls. But we can't say too much about it because, again, it was not detected in our replication cohort. So I wanted to talk a little about malate and how it could potentially be involved in methotrexate-related neurotoxicity. Malate is an unsaturated dicarboxylic acid pictured here. It is speculated to deplete glutathione, which is an antioxidant causing acute renal failure, oxidative stress, and lipid peroxidation. This group also did a study on how lipid peroxidation actually can be associated with neurotoxicity, and malate enhances the formation of free radicals and peroxides, which might interfere with the cell's ability to eliminate these highly reactive intermediates, which can culminate oxidative stress and lipid peroxidation. So of course, we need to do more research on this and do these analyses in a larger prospective cohort. But for now, it is an interesting biomarker of cognitive damage that we could potentially look into that is detectable in patients prior to the clinical manifestation of neurotoxicity. And I don't want to get too much into the proteomics analyses since they are very new, but there were 27 significant proteins that we found doing the similar analysis, and here they are. So unfortunately, I wanted to look into these proteins and kind of do a gene set enrichment analysis, seeing if the metabolites and proteins were in the same similar pathways or the gene ontology, just to see how they're connected, which I didn't get around to doing that yet since this is very new, but I'm going to add it by the end of this summer, just to see. That would again provide another level of validation to results, because if the metabolites and proteins are tied in pathways. So say malleate and one of these proteins are in the same pathway, we can see that even with our small cohort of patients, that these results might be of significant interest. So I just wanted to end with some concluding thoughts. The current standard for care of methotrexate neurotoxicity is to stop or halt treatment, which can jeopardize treatment outcomes or even increase the risk of relapse. So it's not a very proactive approach. Metabolomics and proteomics can help provide a more proactive approach by, if we take a sample of CSF, we detect one of these biomarkers, we can stop or prevent the neurotox happening before it does, rather than wait until it happens and then jeopardize treatment outcomes. And I just wanted to wrap up by saying this study is part of a larger research consortium from Baylor College of Medicine titled Reducing Ethnic Disparities in Acute Leukemia. And I started working to do more of the prospective studies at Chalk Children's Hospital, since that is one of the hospitals enrolled on this study on ReDial, the Reducing Ethnic Disparities. And they just have so much work in store for them. And I'm excited to hopefully share with you guys more work on what they do as it comes along. So yeah, I'm happy to take any questions and thank Chapman University and Schmidt College just for an amazing experience, inspiring us to do these amazing programs and building us as scientists. So yeah.
Thanks, Edena. And that worked out beautifully, given we have overcome the technical challenges here perfectly to learn more about pediatric cancer biology. The floor is open for questions. We have plenty of time, and you're welcome to put them in the chat or raise your hand in the air. I see maybe Dr. Basafi is chomping at the bit.
Edena, that was absolutely fantastic. Thank you for that talk. I know that about a week ago you reached out to me, or you had a question for me about combining the two data sets, the proteomics and the metabolomics. Question, I believe the question was, is there an overlap, right? And
right.
If there is, can it be, how would you find out if there is an overlap? I realized I wasn't much of a help when I told you that there's software out there that can do that. Did you find out something?
I talked to my PI, and like he obviously he knew stuff, but I kind of just wanted to come with him with something rather than be like, I have no idea how to approach this, because like we did in molecular genetics, that was more of a gene-based thing, so it made sense. But I was kind of confused on how to write a metabolite, because is that associated with the gene name? Do you know what I mean? So he does have a workaround. We're gonna work on it this coming week. Unfortunately, I just didn't get to it before the presentation, but I will be including it in my paper if you read it.
Okay, good. And just a follow-up question. I hope I didn't miss that. The data that you presented, how many individual data sets does that represent? How many individuals sample sets stand behind or are behind that?
Do you mean the like sample size, or?
Yes.
It was about, if we go, so the discovery cohort had 12 cases and 22 controls and 14 cases and 29 controls for the replication. So it was a very small sample size, but that's the exciting thing about this study, that now that they got that fund to do this like research consortium with more hospitals, this was only Texas Children's. And it's not that neurotoxicity is so common in all pediatric cancer patients, but I guess what inspired their study in the first place was they published another paper. A lot of the clinicians that work at Texas Children's Hospital were noticing that patients with Hispanic ethnicity had a higher incidence of cases of neurotoxicity, and that was further associated with increased risk of relapse, and just because of the changing the treatment. And of course, if you go off the protocol, it's not going to be as efficient. So they're gonna replicate this. Yeah, so I'm excited to see where that goes, because yeah, of course this was too small of a sample size.
Thank you.
Other questions from the audience?
I'll ask one. I thought that was another great presentation, and I think we're not doing our jobs if we don't call out how amazing it is when a student can recover from technical glitches, which you should never apologize for, even though you did. So don't do that again. And then give that presentation. That's remarkable what you just did. So kudos. Yeah. I actually have a question buried somewhere in here. So it's interesting to me that the number of sort of significant markers that you pulled out was much, much greater for the proteome than it was for the metabolome, right? 27 versus 8. How do I feel about that? Is there a biological sort of reason why we would expect proteins to be more responsive than the small kind of metabolites, or is that just dumb luck that you got 8 and 27?
Well, to me, I just think probably metabolites that's the most representative, right? So if we have a different, even going back to genomics, like if you have a different gene, well, who's to say that gene difference is actually expressed and changed? So I think that maybe, yeah, the proteins are different, but also if you go back to even though the P values were better, the directional effect weren't as significant, I think, as the ones for metabolites are more like 1.3, like 0.6. These are like 0.99 or 0.98. Is that really different? Who's to say? So I think that is one reason, and also because metabolites are, I don't, as the most representative of what is happening in that body at that specific moment. In the discovery and replication cohort, one of the CSF samples were collected plus or minus 90 days to an event. So that was not a great study design, in the sense that 90 days after is not helpful for us, right? We want to have it be 30 days prior is a much better marker, because that is more representative of, is a neurotox event going to happen clinically, and we are using that data. But this one wasn't as stringent of a, like, I don't know what to say, but it's not as stringent of a threshold, I guess. So I think that is another reason for that issue. So.
Great, thank you.
Edena, you have one last question in the chat, but in the interest of time and keeping us on schedule, lest Dr. Schwartz become angry with us, I want to congratulate you on a great presentation, and as well as a great time at Chapman and off campus as well. And we wish you all the very best for all that's to come. And we have our last presentation now of the morning. Cailyn Sakurai is we're gradually going to get bigger and bigger in scale here. And now we're also switching study systems to CHIA. Cailyn's going to talk about the identification of genes involved in CHIA PAMP-triggered immunity. She's finishing her bachelor's in biochemistry and molecular biology, and she has a minor in psychology. Cailyn, the floor is yours.
Thank you so much. So hi everyone, my name's Cailyn, and for my capstone, I work to identify the genes involved in CHIA's PAMP-triggered immune response. So CHIA is part of the mint family and is widely known for its seeds. The seeds have a balanced and near complete nutritional composition profile consisting of dietary fibers, antioxidants, vitamins, minerals, and also omega-3 fatty acids. Most importantly, chia oil is very healthy in comparison to other commonly used oils due to its high concentration of omega-3 fatty acids. And so due to these nutritional properties that chia seeds have, the demand for its production has been increasing in the United States. And as this demand continues to increase, chia will have to expand geographically, where it will inevitably face issues such as pests and also pathogens. Therefore, it is essential to identify these genes that drive the immune response in chia. So a plant's immune response first works by recognizing what are called pathogen-associated molecular patterns, also known as PAMPs. These PAMPs are conserved domains within pathogens. And so in this figure here, you can see that the y-axis shows the amplitude of defense. And so when plants recognize these PAMPs, this stimulates the PAMP-triggered immune response and causing a higher level of defense within the plants. However, pathogens create or produce what is called effectors. These effectors work to suppress this PAMP-triggered immune response, as you can see by these little circles decreasing the amplitude of defense. However, plants have evolved and they have created what are called these R proteins here, which bind to these effector molecules and stimulate a more robust immune response called the effector-triggered immunity. And so in my particular study, I focused on the PAMP-triggered immune response in chia. So a common example, or one example of PAMP, is called FLG22. And FLG22 is a conserved domain on the flagellum of bacteria. And so I use FLG22 to mimic bacterial infection to activate the PAMP-triggered immune response in chia. So in this study, I looked at two genotypes of chia, one which is Pinta, which has been domesticated for agricultural use, and I also looked at the wild type. And so this is the first study that comprehensively profiles chia's ability to combat bacterial infection at the transcriptome level. And so this study will allow us to, one, be able to understand how chia responds to bacterial infection, and also two, identify the key genes in the immune response of chia. And so just going over a quick methodology flowchart, I first treated my chia with FLG22, extracted RNA, then prepared a cDNA library that was then sent out for sequencing. Upon receiving these sequences back, I determined what were called differentially expressed genes, performed a gene ontology enrichment analysis, and then validated my results via qRT-PCR. So going into what I completed in the lab, so I germinated my seeds on nutrient-rich agar for 2 weeks, and then I incubated them in water with or without FLG22. Following the incubation, I collected tissue that I then used for RNA extraction, prepared a cDNA library that was then sent through sequencing. So upon receiving these sequences back, I determined what were called differentially expressed genes, or DEGs. So what exactly are these differentially expressed genes? So in the schematic here, you can see the expression level from low to high. And so looking specifically at gene 1, you can see a high level of expression in the treatment in comparison to the control. And so this difference in expression is statistically significant, causing gene 1 to be differentially expressed. In contrast to this, if you look at gene 2, you can see that the difference in expression level between the treatment and the control is very small. Therefore, the difference is not statistically significant, and this gene is not differentially expressed. So I determined these differentially expressed genes using EdgeR, which is a package in RStudio. I used the following parameters, which was a false discovery rate of 0.01 and a log fold change greater than the absolute value of 1. So after identification of these differentially expressed genes, I performed what was called a gene ontology classification. So what exactly is this GO classification? So in the schematic here, I will go by the steps to explain it a little further. So looking at a pool of differentially expressed genes represented as different color circles, it's hard to see a trend. What GO classification does is it classifies genes based on their sequence similarity to known genes. So in this hypothetical example, you can see a mixture of DEGs were sorted based on their color, which is analogous to the biological pathway that they belong to. While classification of DEGs are helpful, it does not paint a complete picture. Another useful follow-up analysis is the enrichment analysis that aims to highlight important biological pathways. So what exactly is this enrichment analysis? So if you were to have a biological pathway with 46 genes and a total number of differentially expressed genes of 2,809, just due by pure chance, you would find that 2.5 genes would be present in this biological pathway. However, if in your results you actually find that 15 genes are part of this biological pathway, this means this biological pathway is enriched at a fold value of 6.5, which entails that this pathway is relevant to your experiment. And so after I determined my differentially expressed genes, I performed qRT-PCR to validate their expression. So the appropriate reactions were set up in a 96-well plate and ran through a thermocycler in order to determine the gene expression. I looked at specifically genes within the treatment and the control, and as you can see here, the treatment has an mRNA fold change about 4, while the control has 1. So this means that the treatment induces a 3 times higher expression in this gene versus the control. So now moving on to my results. So looking at the differentially expressed genes in both Pinta and wild type, I saw that there are more upregulated genes, so genes that were seen to be highly expressed in the treatment in comparison to the control, than the amount of downregulated genes for both genotypes. In addition to that, I also saw that there are more differentially expressed genes in the wild type genotype, indicated by the green bars here, in comparison to the Pinta. So this highlights a possible difference in the response to flag 22 between these two genotypes. In my study, I also wanted to look at the similarity between these two genotypes, and so I compared the gene identity between the wild type and the Pinta of the differentially expressed genes. And so what you can see here in this overlap, there is about 48.2% of the genes are commonly shared. However, both accessions have their unique response, in which 11.1% is part of the Pinta and 40.8% is part of the wild type. So to further validate these differentially expressed genes obtained for RNA-seq, I quantified the expression of a subset of upregulated genes via qRT-PCR. And so each dot that you see here represents a unique gene. And so what I found is that there's a good correlation between the qRT-PCR and the RNA-seq, such that when a gene is upregulated and the RNA-seq, it's also found to be upregulated when I perform qRT-PCR. So to further understand the differentially expressed genes that was shared between these two accessions, I performed gene ontology enrichment analysis. And so as you can see here, I found a total of 13 biological pathways that had a direct relevance to the immune response of chia. And so just some notable examples are the defense response to fungus and the defense response to bacteria. To further understand the commonly shared genes between these two accessions, both Pinta and wild type, I wanted to look at the individual expression of each gene. So in this figure, I plotted the expression in the wild type and also the expression in the Pinta. And so as you can, with each dot representing a unique gene, and as you can see is that this graph is skewed slightly to the left, which represents that within these shared genes, there's a higher level of regulation in the wild type in comparison to Pinta. If you look at this particular gene here, you can see that it's upregulated at an 8-fold, while in comparison to Pinta, it's upregulated at a 2-fold. In addition to that, if you look at this gene here, you can see that it is upregulated in the wild type, indicated by the positive log fold change, in contrast to it being downregulated in the Pinta. And so this is a heat shock transcription factor that is involved within the stress response to various plants. So we observed the following pathway. So while we looked at the shared response between these two accessions, we also wanted to look at what was the biological significance of these unique genes for both the Pinta and the wild type. So we observed the following pathways enriched when we looked at the specific differentially expressed genes with each plant. Note that most of these pathways are also present in the differentially expressed genes shared between both Pinta and wild type. Also, you can notice that this biological pathway, such as response to hormone, and then the response to lipid, is also seen in the wild type. So what does this mean? This means that although this pathway is enriched in both accessions, the genes that are responsible for this enrichment are different in the Pinta and wild type. This is an indication that these two plants respond differently when subjected to FLG22. So we essentially observed two scenarios. And with the example of the hormone pathway, there are a number of genes that are commonly shared to the FLG20 treatment between Pinta and wild type. In addition to that, each accession also has a unique response depicted here, which is 39 genes for Pinta and 38 genes for the wild type. For the case of the wounding pathway, again, there is seen to be a shared response consisting of 41 genes shared between Pinta and the wild type. However, an additional genes activated in this wounding response was seen in this wild type and not seen in this Pinta. So this observed difference between the wild type and the Pinta's response to FLG22 possibly indicates that the wild type accession has a more robust and stronger immune response to bacterial infection. This can be possibly exploited to target genes within the pinta to create a more effective response as they expand to new geographical locations and face new pathogens. So the next steps of this project is to perform an actual bacterial infection assay using different bacteria to confirm, in fact, if the wild type is more resistant to bacterial infection, just like our expression analysis shows. In addition, we would also like to do gene silencing to show whether those genes that are shown to be higher expressed in the wild type or unique to the wild type are in fact contributing to the ability of the wild type plants to combat bacterial infection. So thank you. Here are some of my references, and then just a little acknowledgement for Dr. Octavian.
Thank you very much for a really interesting presentation on a really interesting species. And we have time for questions from the audience. You can either drop them into chat or speak up. I will lead and put you on the spot. So some of the defense genes, if we could maybe look at some of the compounds that are generated by these genes, things like salicylic acid, sometimes we select for those defense compounds. We want to breed for things that that plants have evolved over time for purposes of defense. Perhaps most notably, for those of you that know me, caffeine in my cup of coffee here. Are any of the things that you identified also things that are that might be of interest for, say, the taste or the nutritional properties of chia?
Well, not for nutritional taste and properties. We were mostly focused on the immune response. So there are, as you said before, with jasmonic and salicylic acid, there are compounds that can change the taste of it. And so in that case, with what I found, there was a higher upregulation in the response to salicylic and jasmonic acid, which could possibly indicate it changing the composition profile of chia. However, this would have to be further validated through more analysis, just because my research particularly focused on understanding the genes and the immune response. So when I was doing my gene ontology enrichment analysis, I focused on genes that I knew had or were revolving around the immune response to chia.
So we're not sure if the wounding in the, say, green material then also upregulates anything that ends up in the seeds themselves.
Mm-hmm. Yes, exactly.
Okay. Okay. Other questions from the audience?
Let me go back to my results.
I have a question.
Okay, sounds
If
good.
I can. So you said that you did this flag 22 treatment, and these are all the differences that you found in the gene expression, and then you suggested that due to the level of the gene expressions, you know, in the wild type, or higher levels of the gene expressions in the wild type, and also because of some pathways enriched in the wild type and not in the pinta, you said that it looks like that the wild type is going to respond better to the actual bacterial infection, right?
Yes.
So why did you go this way, and why didn't you just infect in the first place with the bacteria, so that you figure out whether in fact the wild type is more resistant than the pinta, even before you do this transcriptome analysis? So why did you leave that for the next step? Now, what if wild type is not more resistant to the bacteria compared to the pinta?
Yeah, that's a good question. So as I previously stated, we use FLG2, which is conserved domain within the flagellum of bacteria. And so this conserved domain stimulates the PAMP-triggered immune response in chia. And so our goal of this study was to specifically focus on the PAMP-triggered immune response. And the idea, if we were to use an actual bacteria to infect our chia, this would stimulate not only the PAMP-triggered immune response, but also the effector-triggered immune response. And since that wasn't the goal of what we were focusing on our study, the addition of using bacteria versus FLG2 would seem to be not as important. So as that, I wanted to focus on the PAMP-triggered immune response. So I used FLG2 rather than an actual bacteria, which would affect other pathways within the immune response.
Okay.
Cailyn, thanks for an absolutely fantastic presentation. Our congratulations on everything that you've achieved for the last 4 years. Our best wishes for the future. I join those in the chat that are sending their kudos to you. So without further ado, it's a pleasure to have Luke Taylor give us the first presentation of the session. He's going to speak to us about clogging by hagfish slime, and he's finishing his degree in biological sciences with a minor in chemistry, and I understand that he's headed back to his home state shortly to begin a degree in medicine. Luke, the floor is yours.
All right, thank you. I'm going to share my screen. Okay, and then just give me a thumbs up. Can you see that?
Perfect.
Okay. All right. Thank you for that introduction, Dr. Goldsmith. I will be talking about my research on the superior clogging by hagfish slime, specifically looking at the functions of the mucus and slime threads. And this is a project that's been worked on at Chapman as well as the University of Guelph. So first, a little background on hagfish and hagfish slime. Hagfish are eel-like deep sea fishes that release thick slime when they're threatened. And there's a picture here of a hagfish, and then here's some slime that someone's holding up that's been deployed in seawater. The slime itself acts as a defense mechanism by clogging the gills of gill-breathing predators. So it attaches to their gills and effectively prevents gas exchange with the water, which effectively chokes the animal and prevents any further attacks. The slime is composed of two main components, being the mucus and the slime threads. And it's extremely dilute, and it's about 99.9% seawater. However, the slime can entrain up to 26,000 times its own weight in water. As a comparison, some of the best hydrogels that we have can only entrain up to 3,000 times their weight in water. So the question is, how is this possible? Here are 3 questions that have sort of guided our research and will guide this presentation. The first is, what is the role of each component? So what do the slime threads do and what does the mucus do? The second is, how good is hagfish slime at clogging compared to other materials? And the third is, how does hagfish slime clog? In order to answer these questions, we conduct experiments with live Pacific hagfishes, or Eptitretus stutii. So we anesthetize the hagfish, and then we electrically stimulate their slime glands in order to remove their exudate, which is basically the purest form of the slime before it's been deployed in seawater. And the exudate contains these packaged microscopic bundles of the slime threads called skeins, as well as membrane-bound mucus vesicles that contain the mucin-like glycoproteins that make up the mucus. And we can use this exudate directly in seawater for our whole slime experiments, which contain both the threads as well as the mucus, or we can remove the skeins and work with just the mucus, or remove the mucus vesicles and work with just the threads. And we'll use these different samples in our drain rate experiments. So in order to quantify clogging ability, we use drain rate. So we have an instrument called an instron that basically measures the change in load or weight of a liquid sample over time as it flows through a mesh. And the way that you can think about this is that something that's really good at clogging and really thick is going to drain very slowly through that mesh and produce a very low drain rate value, because it's going to have a change in weight that occurs over a long period of time. In comparison, something that does not really clog at all, like seawater, is going to flow straight through the mesh and have a very quick change in weight over time and produce a very high drain rate value. So low drain rate means good clogging, and high drain rate means poor clogging. And so we use that drain rate method for our samples under varying conditions. And this here is a summary of the different experiments that I'll be discussing in this presentation today and sort of how they relate to the 3 questions I proposed earlier. The first experiments I'll be talking about are looking at clogging by concentration. So in these experiments, we measured the drain rate of whole slime samples, which contain the mucus and the slime threads, as well as mucus-only samples and thread-only samples at varying concentrations. We've also conducted flushing experiments with seawater, where we measured the drain rate of whole slime and mucus-only samples after being flushed several times with large volumes of seawater in order to sort of disturb their structure and observe the effects. And these 2 experiments relate back to that first question of what is the role of each component. The third experiment I'll be talking about is looking at clogging by other materials. So in these experiments, we measured the drain rate of xanthan gum, hydrated psyllium husk, and a high molecular weight polymer called polyethylene oxide, or PEO, again, at varying concentrations in order to better understand how good hagfish slime is at clogging compared to other materials. And then finally, we conducted changing pore size experiments where we measured the drain rate of whole slime and glycerol samples when we increased the size of the holes in the mesh that we're using. And this is in order to better understand how hagfish slime actually clogs. So our first question was, what is the role of each component? We're going to start with the mucus. What we've discovered is that the mucus component is really what's causing the clogging that we're seeing by the slime. Here on this figure here, we have drain rate plotted on the y-axis. And again, low drain rate values correspond to good clogging and high drain rate values correspond to poor clogging. And this dashed line at the top here represents the drain rate value for seawater. And this is plotted as a function of the total concentration of mucus and slime threads for whole slime samples here in blue and then mucus only samples here in red. And what we see here is that even when we remove the threads and are just left with the mucus, we see that it's able to clog just as well as the whole slime, which contains both the mucus and threads at basically all the concentrations that we've tested. We also know from concentrated thread-only trials that the threads display virtually no clogging ability, and that the mucus really must be present for any clogging to occur. So this makes us think that the mucus is really playing the primary role in clogging rather than the threads. And so naturally the question becomes, well, if the threads aren't clogging, what are they doing? And we've concluded that the slime threads really are there to retain function and clogging ability over time. Again, in this figure, we have drain rate on the y-axis now as a function of the number of flushes with seawater for mucus-only samples here in red and then whole slime samples here in blue. And what we see is after just 1 to 2 flushes with seawater for our mucus-only samples, the clogging ability is virtually diminished and drain rate values return to those that we see for seawater. So basically the clogging performance of the mucus is really diminished after just 1 to 2 flushes. In comparison, whole slime samples are able to maintain their clogging performance and function over several rounds of flushing with up to 7 flushes over time and maintain a significantly low drain rate value. And so this makes us think that the threads really are there to provide structure to the slime that allows clogging and function to endure over time. And this makes sense because the goal of the slime is to remain together and attached to the gills of hagfish predators in order to act as a defense mechanism and prevent further attacks. So the threads really are there to maintain function and structure for the slime. And then our second question was, how good is hagfish slime at clogging compared to other materials? And we've conducted experiments with other materials, and we've seen that hagfish slime displays superior clogging performance over all the materials that we've tested. Again, here on the left, we have mucus-only and whole slime samples for hagfish slime. And then we have xanthan gum, polyethylene oxide, and psyllium husk. And you can just see, based on the nature of the log scale for this total concentration on the x-axis, the stark difference between the clogging ability of these materials compared to hagfish slime. What we see is that hagfish slime can effectively clog at concentrations that are 2 to 3 orders of magnitude lower than any of the other clogging materials that we've tested. That's 100 to 1,000 times better. We also see that hagfish slime can clog effectively at low concentrations that are similar to those that we measure in native hagfish slime, which is around 35 milligrams per liter. And these results are very interesting and have a lot of potential to show how useful and valuable hagfish slime can be as a biomaterial. And then our final question was, how does hagfish slime clog? And this is a really big question, and a question that researchers have been trying to answer for many, many years now. And we still don't fully know exactly how hagfish lime clogs, but we've conducted some experiments in order to better understand how the clogging that we're seeing is occurring in our drain rate experiments. So when we conduct the drain rate experiments, we're using meshes that have holes in them that allow a liquid to flow through. And so the clogging that we see performed by hagfish lime, we concluded could be the result of two different possibilities. The first is that the slime and the threads and the mucus are covering the holes in the mesh and plugging them, thereby preventing any water from flowing through. The other option is that the slime itself is doing something to entrain or entrap the water and resist its flow, so that it's doing something itself. And so to better understand how hagfish lime is clogging in our drain rate experiments, we conducted experiments where we changed the size of the holes in the mesh that we're using. Theoretically, as you increase the size of the holes in the mesh, that it allows for more space for liquid to flow through. And so the drain rate should increase because that liquid is flowing much faster, which is what we see here in these glycerol samples, which sort of acted as our control for these experiments. And I want to highlight that this drain rate here on the y-axis is plotted again on a log scale. So this increase is very significant in terms of the drain rate as we increase the size of the holes. In comparison, hagfish lime is essentially insensitive to the changing pore size. As we increase the size of these holes, there really is no effect on the drain rate that we see for hagfish lime samples. And this makes us think that the resistance to flow that we're seeing is really caused exclusively by the slime via its entrainment of water, rather than just plugging the holes of the mesh that we're using. So in conclusion, our first question was, what is the role of each component? We now know that the mucus causes clogging, while the slime threads provide structure to retain function over time. Our second question was, how good is hagfish lime at clogging compared to other materials? We now know that hagfish lime effectively clogs at concentrations that are 2 to 3 orders of magnitude lower than any of the other clogging materials that we've tested. And the third question was, how does hagfish slime clog? And though we don't fully understand this question, we know now that the slime itself resists flow via the entrainment of water. In a broader context, understanding the distinct functions of mucus and slime threads and how they facilitate the superior clogging performance that we see in hagfish lime may help lead to the development of some unique biomaterials that provide a barrier to flow and then subsequently collapse or biodegrade in nature. In terms of future directions, we're hoping to conduct experiments where we manipulate the slime network in order to observe the effects on clogging and function and better understand the slime structure. And we can do this via cross-linking. So the mucus network contains mucin-like glycoproteins, which form disulfide bonds with each other. And so by adding a cross-linker, we can facilitate this process and bring those mucus threads together and therefore observe the effects on clogging and function. And we can also use a compound called DTT, which effectively does the opposite of this and collapses the mucus network. And then our second future direction is to complete and submit a manuscript to the Journal of Experimental Biology with all of these findings and results. I just want to take the time to acknowledge Dr. Fudge and everyone at the Fudge Lab for all of their support and help on this project, as well as everyone at the University of Guelph. And then I also want to thank Dr. Keller, Kendra, and Brandon for all their help this semester in terms of getting this project to what it is today. Thank you for listening. I'm happy to answer any questions.
Thanks, Luke. We're happy to take questions from the audience. You can either drop them in chat or wave wildly to get my attention or pull yourself off mute, whatever it takes. So Luke, I'll lead here, even though you and I have now talked a lot about pore sizes. I've got a different question for you. I would love to know, so this seems to me to be a relatively effective, if not uncomfortable way to stop a predator from taking a bite out of you. There's not that many cases in my head of something that only evolves once. If it's that good, one often sees that it evolves in multiple taxa and lots of different species. Where else do you see this in nature, either on land or in the ocean? And if you don't see it that much, then why not?
That's a great question. So there are a lot of other species and organisms that produce slime, but the slime that we see produced by hagfish is extremely unique because there really is no other material that's able to entrap water and clog as effectively as hagfish slime. So there are other mechanisms to deter predators, like you can think of squids releasing ink and things like that when they're attacked. So there are other methods and other organisms that do produce slime in a defense mechanism sort of way, but it is very surprising to see how effective hagfish slime is compared to these other materials. So in terms of why other organisms haven't adapted as well as hagfish in producing such an effective defense mechanism, I don't have the answer for that, but we do see similar defense mechanisms and production of slime in other organisms, just not to the degree of effectiveness necessarily that we see in hagfish slime with respect to these specific properties of clogging and water entrainment.
Yeah, it's quite remarkable. That's really remarkable. I've got I think a hand from someone, but I'm not quite sure who it is. You're welcome to take yourself off mute and ask away.
I don't know if somebody else also has their hand. So my question has to do with, so the slime works really, really well. And you showed, over time you were washing it multiple times and it was hanging in there. So when a predator gets slimed, what happens to the predator? Does the predator, over like 5 minutes, will the predator be able to come back? Does it just give the predator, the hagfish time to get away, or what's the time frame for the poor predator in this case?
That's a great question. That's actually something we're trying to figure out. So we know from video evidence of sharks and other organisms attacking hagfishes that this process happens almost immediately, and you can physically see the predator retract from the hagfish and kind of freak out because it's being choked and it doesn't really know what to do, but the predator will usually leave the frame. So we don't actually know what happens to this predator after they're slimed by a hagfish. So we don't know if the interaction is fatal for them or if they're able to remove the slime from their gills. We know that some fishes may employ back flushing in order to rid the slime from their gills. And so these flushing experiments that we did were just flushing the slime. We weren't using back flushing. So we see that the slime is able to endure over lots of regular flushes, but we don't really know how long it'll last or if other methods like back flushing might be more effective at removing it. So we don't have an answer to that question yet. And we really would love to see some future experiments or studies looking at hagfish predator survival following a slime by hagfish, because we really don't know what's happening to these poor sharks that are biting these hagfishes. But it really is an interesting future direction that hagfish researchers might take. So.
Yeah, or just thinking about, when you're thinking of potential uses for this, like how permanent or temporary is it? Interesting.
Right.
Thanks.
Mm-hmm. You're welcome.
Very briefly, Luke, before we transition, do they produce slime at a fast rate? So how quickly can they, I think Dr. Ellertson is sort of asking in the chat, how quickly can they ready themselves once again?
Oh, okay. After they've already used their slime? That's a great question. So it's a local response. So usually when they're bit, when we conduct experiments, we usually empty their glands, and it can take up to a month for these glands to refill, but they have many, many glands lining both sides of their body. And so it's very unlikely that one attack will empty every single gland. So realistically, it takes some time to refill the glands, but if they were to be attacked by another predator, I would say that it's likely that they would be able to slime again and produce a defensive response. So it does take a while to refill, but they're very effective, even with very small amounts of the slime, at defending themselves against predators and choking them. So, yeah.
Luke, I join all the other folks in the chat that are offering their congratulations and their comments on how great your presentation was, as well as our congratulations on everything that you've done over the last 4 years at Chapman, and our best wishes for your transition back to the islands. And with that, we'll transition to our next speaker.
Thank you.
Thanks, Luke. Sanika Pandit is in the midst of finishing a double major in biological sciences and computer science, and it's a bit of a shock for me to see her here for a senior presentation, since I think that she was the first student I had on the first day that I was ever at Chapman. She's going to talk to us today about synthetic platelets derived from designed microgels. And Sanika, the floor is yours. We're almost there. That looks good. And now we just need you to unmute.
Sweet. Thanks, Dr. Goldsmith, for that great intro. And before I get started talking about basically synthetic platelets, I thought we should really first start off by understanding just what our normal platelets do and how they sort of work to, heal a wound. I can basically assuredly say that, all of us have gotten a cut and, bled, and lucky for us, our blood clots. And at the crux of this process are platelets. And platelets are basically, cell fragments that have the ability to mitigate wound healing. And platelets basically do three things. They're able to bind to the site of an injury that we see like right here. So, when we get a cut, platelets rush to the site of the injury. They're able to spread. So at the site of the injury, platelets begin to lay down a fibrin mesh and they spread out to basically plug the wound to stop blood flow. And finally, they're able to contract. So platelets are able to generate an internal force and they draw upon the edges of the wound to basically close it shut and basically help promote wound healing. And from this, we know that platelets need to be three things. They need to be small. Platelets need to be able to, reach the site of the injury without, through the bloodstream. Two, they need to be squishy or highly deformable. Platelets need to be able to basically, both expand and plug up the wound, but also contract and draw the edges of that wound together to close it. And then finally, you need platelets to bind specifically to fibrin. You don't want your body forming clots elsewhere, like not at the site of the injury. So you can imagine the inability to properly form clots can lead to some serious problems. And in order to address this need for platelets in trauma application, the Lion group, along with several other collaborators, has developed platelets substitutes that we like to call platelet-like particles, or PLPs for short. So moving on to our PLPs. Our platelet-like particles are made from an ultra-low cross-linked microgel, or ULC microgel. A microgel, before we can talk about microgels, we need to first understand what a hydrogel is. A hydrogel is basically a polymer network with containing water. And a microgel is just a hydrogel on a smaller scale. Microgels tend to have diameters ranging anywhere from several hundred nanometers to a couple micrometers. And you can see that we start our synthesis off with some pretty simple monomers. We use poly-N isopropyl and acrylic acid, and through a polymerization precipitation reaction, we're able to synthesize a really small ULC microgel. And from the atomic force microscopy data, we can really see that the diameters of these particles are about 1 micrometer. More than that, in this dehydrated state, they're just a couple nanometers thick. And from this, we know that these particles are so waterlogged and so squishy that when they're dehydrated, they're so thin. And at this length scale, the microgels are on the scale of cellular materials and are biocompatible. So at this point, we know that we've made some pretty small squishy particles. And what do we need these particles to do? Well, basically, we need our PLPs to be able to bind to the fibrin network and, spread out and, get in contact with that mesh. But through the process, we also need them to be able to collapse and deform and shrink and, help promote that clot contraction. But at this point, when we've synthesized the ULCs, we won't be able to have that specific fibrin binding. And to be able to get to this point where our particles bind specifically to fibrin, we have to do the second part of the synthesis. Because at this point, our ULC microgel is small, it's squishy, but it's not binding to fibrin. So we tag our ULC microgel with a fibrin antibody that makes it bind to fibrin. But at this point, not much can be, learned about the interactions of the PLP and a clot because we can't visualize it. So in order to be able to visualize it and really understand those interactions of, the PLP and the clot, we tag our particle with a fluorescent dye that lets us really visualize the interactions of the PLP and a clot under a fluorescence microscope. So what's really interesting about these particles is that some seemingly simple materials have been used to mimic an intricate biological process. I mean, the line group in clinical applications has shown that these PLPs can be used to help promote clotting and help promote wound healing. And my goal was to really, for this research project, was to understand how these PLPs interact with fibrin clot. We know they work, but what's that underlying mechanism of why, such simple monomers can basically mimic a complex biological system like platelets without having that inner mechanism. And to be able to do that, I focused on using the super resolution mode of the confocal microscope to really understand these interactions. So unlike a traditional optical wide field microscope, a confocal microscope uses a laser to basically excite a point-by-point slice of the sample. And the resulting image comes from only the in-focus plane, so any off-target noise is basically selectively removed from the pinhole. And by using the super resolution mode of the confocal microscope, we're able to boost the resolution and the sensitivity of the microscope to get a better image of our sample. The AriScan detector basically allows us to get a higher resolution in the X, Y, and Z direction. Here we can see how the AriScan detector significantly improved the resolution of a sample. Additionally, with the AriScan detector, we're able to take Z-stacks, which are basically slices of our sample and then stack and then through depth. And from that, we're able to get a 3D rendering and get a deeper understanding of whatever sample we're looking at. Especially, we're able to get more qualitative and quantitative data about how our particles in particular interact with fibrin clots. So I did a set of experiments where I created a fibrin clot and I took some Z-stacks, so a couple slices through the fibrin to be able to really understand the three-dimensional structure. So on this left video playing over here, you're seeing it play through each of the different stacks that I had taken. On the right, we see a total intensity projection. So basically, it took the three-dimensional model, took all those slices and smushed it down into a two-dimensional figure to really give us a better understanding about the detail of how this clot was formed. Just like I did 3D analysis for my clot, I did some 3D analysis for the PLPs. So again, here on the left, we can see a video working through the Z-stack or the images that I'd taken for my platelet-like particles. And on the right, we see a 3D rendering of these platelet-like particles in space. And by using the confocal microscope, I was really able to get some pretty cool images that told me a lot about how these particles interacted with the clot. So basically, in an effort to determine the interactions of the PLPs in a clot, I ran a whole bunch of experiments. I first started off by making clots by combining fibrinogen and thrombin. And then I added my PLPs to the clot and imaged these at a whole bunch of different time points. So in the first column, we can see my platelet-like particles, and we can see that they're just about, a micrometer in diameter. And moving on, I did experiments where I put them in the clot and visualize them at different time points. So, at 20 hours, we see that they've significantly changed in their shape. In this, at this time point, they're stretched out, they've become elongated, and they're binding to different parts of that fibrin mesh. And then less than 2 hours later, we see that they've changed morphology one more time, where they look smaller, denser, and much smaller than even at time point 0. And they've, pulled together pulled the fibrin together and created or allowed for the clot to basically collapse. And, by doing this, we're really able to visualize how these particles that we made from, again, relatively simple materials are mimicking a complex biological function and pose as an opportunity to serve as an alternative biological therapy. Aside from my initial studies, I also ran a second set of experiments where I used 2 fluorophores. I first had my PLPs dyed, and then I also had the fibrinogen in the fibrin clot dyed as well. And these were some pretty challenging studies. I'm not gonna lie about that. Getting the PLP and the fibrin clot to be within the same focal plane or at the same Z level was insanely difficult. And for the goal of these studies was not only to just understand how the PLP responds to the clot, but see how the fibrin itself responds to the PLP over time. So for these studies, I took another Z-stack of around 35 slices, so at 35 different focal points. It took an image of the entire plane, and I went through all the images to basically process them so the PLP was in the correct intensity, and then the fibrin was in the correct intensity, and then go through all those 35 slices to find those, 1 or 2 slices where we can see the platelet and the fibrin interacting together. And then this sort of leads me to future directions. I think it took me quite a while. The first time I was, running these experiments, it took me nearly a semester to be able to synthesize a platelet-like particle. And now I can run several experiments through the semester. So moving forward, I hope to be able to run more of the experiments where I have my PLPs in the labeled fibrin clot, so I can get more qualitative and quantitative data about how these PLPs are interacting with the clot. And I think I would really like to thank Dean Lyon, Dr. Islam Della, and the Ghosts in the Confocal Microscope for really supporting me through my research endeavors, and Dr. Keller for inviting me to speak at this and really challenging me to really put myself out there. Thank you, and guess I'll take questions now.
Thanks, Sanika. I think, the obvious first question here is going to be, there's going to be a lot of people that are wondering about the ghosts in the confocal microscope and exactly what those look like. Are happy to take questions from the audience. You can either pull yourself off of mute or feel free to type something into the chat. I do have it on good authority that these are incredibly challenging images to take. Could you tell us what is, just so that we have some sense of this, what is the depth of a Z slice? So when you change from one Z to the next Z, what's the scale of that change?
I mean, for my PLP images, this was like only maybe a couple hundred nanometers to really get into the Z of it, because I was trying to get in focus for just a single PLP and I happened to find more, and I was like, well, that works out for me. For the 35 slices that I took, which was this one. Oops. This one, I think it ended up being, a couple, I'd say it was close to a couple hundred micrometers, just because they were definitely not in the same focal plane. They were long ways apart. So I had to make the Z-stack bigger, but in the intermediate images, they were just black because there was no fluorescence, because there was nothing labeled in those intermediate slices. So it took a lot of processing and compressing the layers down to be able to get, the PLP and the fibrin to be on top of each other for this final image.
There's a question from Cailyn in the chat. How do you think the PLP makes the clot contract?
I'm not too sure about that, but what we hypothesize is it uses like a mechanism similar to like a Brownian ratchet, where the PLP will come in, bind to the fibrin, and then contract it a little bit, and then rearrange, and then bind to the fibrin again and contract it, and then over time we get this overall like clot collapse. But that's my best guess on that.
Other questions from the audience?
I have a question. Here you already showed nicely in this slide that PLP and fibrin are with this overlaying images that they are nicely binding. And then you said that now you're more efficient in this work, you're going to do much more experiment. Can you describe specific 1 or 2 experiments? I'm just curious, what else can you do in order to better understand this whole mechanism? Because you already showed that they bind and contract. I'm just wondering, what else? Can you give me like 1 or 2 specific examples of experiment that you're planning to do?
Yeah, I think I can say that I was, let's go back a couple slides. I was pretty efficient in being able to really visualize these PLPs and see how they clot and contract, but I can honestly say the way that I make my clots may not be the most efficient. I make them in a microcentrifuge tube, and I think that's one of the main reasons why, I make them in the microcentrifuge tube, mix them around, and then pipette them onto the slide, and that's what leads to this like big gap in the Z direction. So being able to get a really good understanding of how the clot itself with the labeled fibrin and the PLP interact is something that I want to, look into. So optimizing my methodology to, really get these PLP and fibrin within that, couple like same range, so I can actually be able to see this PLP contraction expansion within the labeled clot is what I sort of meant with that in terms of future direction. Because I know it works. I mean, you guys are just taking my word for the fact that there's fibrin in here, that there's a clot, but none of us really can see that. And being able to delve a little bit more deeper into that is what I'd move further with.
I think we have time for 1 more question from the audience.
I'll ask one if no one else has one. So Sanika, I want to go back to your sort of very first synthesis step where you talked about how you make these PLPs to be both small and squishy. And I'm just curious, can you sort of change the recipe that you use to make them smaller and more or less squishy? So if you didn't use NIPAM or acrylic acid and you mixed other things together, do you get different characteristics of the ultimate microgel or the PLP that you create? And is that something that people are thinking about, right? Is there sort of a balance between smallness and squishiness that you can play with by changing the recipe of these PLPs?
I mean, honestly, kind of outside of the scope of the project, because I just took the line group's, word for it that these PLPs work. But, I'll take my best stab at this. But I think that, you always want to make something a little bit smaller, a little bit more squishy. I think smallness is like the big thing, because with particles, that are foreign to the body, you don't want them getting stuck in your body, especially in like key parts of your body, like the kidneys. And if you make these particles as small as possible and as squishy as possible, you'll be able to, filter them out through your body, like, naturally, and not cause, further damage to things that you don't want, organ damage. You want to stop a clot, but you don't want to go into renal failure. So I think that's always a goal, but again, not particular in the scope of the work that I've been doing.
Thank you.
Sanika, I'm joining everyone in the chat and all of those hand clap emojis that I've seen coming across in an absolutely fantastic presentation and in everything that you've done in the last 4 years. I know that we get to hold on to you for just 1 more semester. And so we're wishing you the best for the summer and that semester and all that is to come. Thanks.
Thank you.
We are changing scales and disciplines here for this next talk. It's a pleasure to have Kendra Ellertson, who's finishing a degree in biological sciences with a minor in environmental sciences, and she is going to talk to us about seeing light from a different angle and the effects that it might have on plant function and growth. There are many things that I could probably say to deeply embarrass Kendra, but I will try to restrain myself from doing so. And Kendra, the floor is yours.
Thank you, Dr. Goldsmith. Let me just share my screen. You guys can see this okay?
Perfectly.
Okay, so I'm going to be talking about the project that I've been working on for the past year. I've been looking at the effects of diffuse light on plant function and growth. So what is diffuse light? When we go outside on a clear sunny day, 100% of the light that is coming to Earth's surface is direct, meaning it's in these straight parallel beams that are hitting the Earth's surface and the plant surface in kind of this parallel direct fashion. But when diffuse light happens, it's light interacting with atmospheric particles that scatter the light. So instead of light coming down in a straight line, it's coming in from all angles. So we think that because this light is now hitting a plant in all angles instead of just one, that this could increase its productivity and growth. So diffuse light in nature, ecologists started to really think about the effects of diffuse light after the eruption of Mount Pinatubo in the Philippines. This was a massive eruption that blew a ton of aerosols and atmospheric particles into the air that created a diffusion of light. So all of these particles that were blown into the air are scattering and refracting this light, but this phenomenon was seen globally, so not just where this eruption happened. There was also a global drawdown of CO2. And when ecologists started to think about why this was happening, plant productivity came to mind. So if there's a global drawdown of CO2, that could mean that plants are using more carbon, meaning they're being more efficient, doing more photosynthesis. So global climate models suggest that there's going to be an increase in diffuse fraction in the coming years, meaning an increase in water vapor, atmospheric particles, aerosols, all which refract and scatter the light, making it diffuse. So we need to understand how this difference in the angular quality of light is going to affect crop and vegetation growth. And that was kind of our leading question going into this project, is how does diffuse light affect plant physiology, morphology, and growth? And we hypothesized that diffuse light would increase photosynthesis and therefore growth, while potentially reducing water use. So a little bit about the project and how I actually tested this. I actually did this project during COVID in my side yard in Orange, California. And how I did this is I built and constructed 8 open air greenhouses. So they had ambient atmospheric conditions. The only thing that was different was the light quality that was coming in from the top. So half of my greenhouses just had a clear glass panel for direct light, and the other half I sprayed with a diffusion paint to scatter the light to about 50 to 60%. And I used adjustable greenhouses so that I could move the top up and down as the plants grew to make sure that the plants were receiving the same amount of light quality throughout their entire life cycle. And I use tomatoes because tomatoes are an important worldwide crop. They're grown around the world, and we wanted to see how this important crop, would do in different light qualities. And I used Roma tomatoes because they're a determinate variety, meaning that they're all going to flower and fruit around 60 to 75 days, making it really easy for me to quantify differences in growth between the treatments. So I took a ton of measurements over the course of this experiment, looking at physiology, growth, morphology, functional traits, structural traits, but I'm only going to focus on a few today. I'm just going to focus on how light quantity affected temperature, biomass, and ambient photosynthesis conditions. So first here we have just a graph looking at how much diffuse light was actually in my greenhouses. So on the x-axis, we have time of day, and on the y, we have diffuse photosynthetically active radiation. So my smooth lines represent my direct greenhouses, and my dotted are my diffuse. And this is just showing that throughout the plant's life cycle, there was significant diffusion of light in my diffuse treatments. So basically the treatment worked, and I had significant diffuse light throughout my treatment during the plant's life cycle. So how did this diffusion of light affect leaf level plant physiology? Here we have box plots of my different treatments. So the light is direct and the darker color is diffuse. And we're looking at ambient photosynthesis here. And photosynthesis is measured with an infrared gas analyzer called a LICOR, which basically just clamps a leaf into a chamber and measures the molecules of carbon and water that are coming in and out of the plant to measure photosynthesis. So after month 1, there were really no differences in photosynthesis. As you can see, the box plots are pretty much the same. But after month 2 is when we actually started to see some quantifiable differences. But we actually saw the exact opposite of what I hypothesized. So I thought that because diffuse light is coming in at all these different angles, that would increase plant productivity and increase photosynthesis. But I found that direct plants actually had higher photosynthesis. And I used t-tests to do the statistical significance here, and this relationship was significant. And this same pattern of nothing really happening after month 1, but starting to see some drastic changes after month 2 was true for other physiology measurements as well, like transpiration, stomatoconductance, and intrinsic water use efficiency. So moving on to like the whole plant level now, these are box plots of my relative growth rates between my treatments. So again, the light colored box plots are direct, the darker diffuse. And I looked at height, stem diameter, and leaf count. And as you can see, the box plots are the same. So diffuse light had no, did not alter growth at all. They're pretty much the same. It didn't do anything basically. But we did see a curious result with whole plant biomass. So at the end of the experiment, I cut down all the plants, put them in a drying oven, and measured how much biomass they had. And we saw that the diffuse plants had almost double the amount of biomass than the direct. But after kind of going back through the data and looking at pictures of the plants that I had taken throughout, we actually think that this is due to phenology. So the diffuse light or the direct light treatment was a little bit hotter. These plants started yellowing, shedding leaves, and just dying a lot quicker than the diffuse plants. So we think that there's maybe some phenology story to this, but I don't have a lot of data on the actual senescence because it kind of surprised us. We weren't really looking for that. But we think that this difference in biomass might be due to changes in phenology from maybe a higher temperature in the direct greenhouses. So here we actually have what temperature I had in my greenhouses. So on the X time of day and on the Y, the temperature in Celsius. So again, my smooth lines are direct and my dotted are diffuse. And we found something kind of cool. We found that light diffusion actually decreased the greenhouse temperature by about 2 to 3 degrees Celsius. And this also translated to a 2 to 3 degree drop in leaf temperature as well. And this is cool because tomatoes are grown in large scale greenhouses, and those large greenhouses take up a lot of energy to cool. So with this diffusion, being able to cool the greenhouses by this 2 to 3 degrees, we have the potential to decrease the energy requirements needed for these large scale commercial grows. So just a summary, again, even with this 50% increase in light diffusion that I had in my treatments, there was no effect on plant physiology or growth. And we actually saw the opposite of what I hypothesized. So we saw a decrease in photosynthesis. And then we also saw that increase in biomass in the diffuse plants, but again, we think that might be because the direct plants started to senesce and shed leaves a lot quicker. And then with that 2 to 3 degree drop in greenhouse temperature and leaf temperature, we have the potential to decrease those large scale energy requirements needed for greenhouses. So stepping back out a little bit more, why is this important? I told you at the beginning that there's going to be an increase in diffuse fraction in the coming decades, either from aerosols or cloud cover. And if we plan to be resilient in times of a changing climate and continue to feed our planet, we have to understand how plants are going to respond to changes in various light environments, not only ambient, direct and diffuse conditions, but also in artificial greenhouse settings as well. So what's next? When I started this experiment, I wanted to quantify fruit quality and fruit yield of the tomatoes. But since I live in Southern California, I had a rat problem. And the rats basically ate all of my tomatoes. So I had to kind of reframe my question and think about how I could, change my project a bit. But in the future, I think it would be really interesting to, when there's not a rat issue, to look at how this angular quality of light is affecting, the fruit quality. So color, firmness, nutritional quality, and yield. And also how this diffuse light is changing water use. I did look at intrinsic water use efficiency and whole plant water use efficiency. So just how effectively these plants are using water. But I think, diving a little bit deeper into this, especially in the face of a hotter and drier climate, we want to know how these plants are going to respond. And since I saw this kind of curious result in senescence in my direct plants, looking closer into how seasonal changes and phenology are changing, these plants. And then just, since we're looking at how crops and vegetation are going to respond, how can we improve greenhouse design for large scale crop grows? So that's it. Thank you very much. I'd like to say thank you to my friends for helping me with these plants and assistance in the field, as well as Dr. Keller, Luke Taylor, Brandon Bernardo, for just constructive comments and really helping me throughout this whole process. And Dr. Berry and Dr. Goldsmith for being my mentors and supporting me for my time at Chapman. So thank you. And any questions?
Thanks, Kendra. We have plenty of time for questions from the crowd. You're welcome to drop them into chat or come directly off mute as you see fit.
I have a question. When you created this diffusible greenhouses, and you showed that nicely with a figure showing how diffused the light is, did you by any chance look at the quality of the light underneath? In other words, the spectrum of light, was there like a significant difference? Because I'm worried that with the process of diffusing the light, you're basically messing around the normal spectrum of the direct sunlight.
Yeah, so we used a light meter called a sun read meter that was in my greenhouses at plant height the whole time. So we were measuring light quantity and quality the whole time. And unfortunately, I saw a little bit of a reduced, light quantity in the diffuse greenhouses just from like a little bit of a shading effect. And we think that maybe that reduced light quantity might have, affected our photosynthesis measurements. But overall, with also other diffusion materials, they've seen that, there's potential to not decrease that light quantity, but it depends on like what materials you use and how diffuse it is and stuff.
How about the quality? So I'm more interested about the quality, not the quantity. So when you plot the spectrum of the light between red, green, and blue, do you see like any significant difference in the peaks of the red, the blue, and the green parts?
Yeah, we actually didn't do any, spectra quantification, but I think that they would be relatively similar.
Well, I would test that.
Yeah, it'd be an interesting, I definitely, that's something to look at too, is how the how plants are using different types of light, not just angular quality.
Yeah, because it might help you, answer some of the, or explain some of the interesting results that you found through.
Dr. Fudge, I see a hand raised.
Yes, Kendra, really nice job. Interesting study you've done. I'm interested in these Lycor photosynthesis measurements and how they're done. Is there a little light inside the Lycor that illuminates the leaf, or are you using the natural light when you make those measurements?
Yeah, so for these measurements, I'm using natural light. So with the Lycor, it's like this little chamber almost, and you clamp the leaf in it, and then on the top, it's just a clear piece of plastic. So we're just getting ambient sunlight. We also did some measurements testing how these plants would respond to, if the plants were grown in direct light, how they would respond to diffuse light or changes in light quantity. But we found that they, were relatively the same. They don't care. The light that they're grown in is what they like the best.
Mm-hmm. Can I just do a quick follow-up one?
Yeah.
So I'm also curious about the ecological implications. So obviously agriculture is a big, implication for this, but what about plants that are living under the canopy and maybe they're getting light that's bounced around a lot by the time it gets to them?
Yeah, we know that diffuse light is really good at scattering light. And when we have direct light, we get kind of these like high sun spots and shade spots in the leaf canopy or in the plant canopy. But with light diffusion, it's just a lot better at spreading this light out throughout not only the plant canopy, but the leaf surface. And so because of that, we just think that especially leaves on the lower part of the canopy are still being able to receive a lot of light instead of, indirect light where potentially they could be completely shaded. So it has potential for, illuminating a lot more leaves instead of just ones at the top.
But would you say that the ones at the top are getting way more direct light and the ones, there's like a gradient of diffusiveness?
Yeah, I would say that the plants or the leaves at the top are definitely getting more, but since we have this light coming in at all angles, it's also able to come in from the sides, not just from the top. So we get a more even spread of light throughout the whole canopy.
Gotcha. Thank you.
We have time, I think, for one more question from Dr. Scherf.
So the previous question might have somewhat answered it. So my question has to do with, so just to clarify the idea, if you have this diffuse light, you're getting, the idea is you're going to increase photosynthesis because you're hitting more surface area of the leaves. So you're getting more photosynthesis that way. But also, you showed the photo at the beginning with, you have a big volcanic eruption. I'm thinking that that's going to block a lot of light. So if you have a lot of particles, you're going to diffuse the light, but won't you also be screening some of it? So what you're looking at is going to be some sort of function of how much light you're screening out versus how much you're letting bounce around. So it might be a little complex.
Totally, yeah. The example of the volcano might be a little bit extreme, but on a day-to-day basis, we can think about just cloud cover. Like when you're outside, you're gonna put on sunscreen when it's a clear day, but you might not think about it on a cloudy day. You're still getting the same amount of light. It's just a different type of light. It's coming in at all angles. It might be, blocked a tiny bit, but you're still getting all of that light. It's just coming at you in different angles instead of right from the top.
Okay, yeah, I've never put one arm out in the direct sunlight and one under sun cover with the same amount of sunscreen, and so one gets burnt more. So I guess that was my question, what that function is. But okay, got it. Thank you.
I know we might have another question, but I want to keep us close to time here. Kendra, thank you for a really great presentation and for 4 really fantastic years of allowing yourself to be subjected to everything that was going on in the Goldsmith lab. I'm really very excited to see all the cool things that you do in the years to come. Our final presentation by my notes here for this session and this day is from Brandon Bernardo, who is finishing up a degree in biological sciences and a minor in philosophy, and he is going to talk to us about some work that he's been doing on spatial and temporal patterns of water use efficiency in the forests of Switzerland. And Brandon, the floor is yours, and we can see your slides.
Thank you, Dr. G, for that fantastic introduction. So like you said, I'm Brandon Bernardo, and I'm going to be talking to you guys a little bit about forest water use efficiency patterns across Switzerland. So first to begin, I just want to kind of define what water use efficiency is. And so the textbook definition of water use efficiency is CO2 gained through photosynthesis divided by H2O loss to transpiration. This is an integrated measurement, and it gives us insight into plant and also ecosystem function. So it's a really important measurement for us. However, it's traditionally very difficult to measure. It requires strenuous on-site measurements of individual leaves or whole plants. So this really limits our ability to measure water use efficiency across really large spaces or across large time periods, just because of the strenuous on-site data collection that it requires. However, for my project, we utilize NASA's novel EcoStress instrument on the International Space Station, which is essentially just a really large camera on the side of the International Space Station. It circulates the planet in orbit with the space station, and it's consistently taking 70-meter resolution images of the planet. It also takes measurements of temperature as well as measurements of greenness from the images. And from those two measurements, it's able to provide an estimate of water use efficiency, all from the International Space Station. And utilizing EcoStress, we decided to study water use efficiency in Switzerland because we believe it's an ideal location due to the variability of the site characteristics across the country. So you'll see here, the Alps kind of are a really main factor of Switzerland that allow us to look at really different environmental conditions. So that the elevation increases, precipitation varies widely, as well as the temperature. And so using all these really wide range of environmental factors in a relatively small area allows us to look at water use efficiency and how it varies with these different environmental factors. Additionally, in Switzerland, there are relatively few dominant tree species. So oak, beech, and spruce are the three main dominant tree species across Switzerland. These three species inhabit their own unique environments that also aids us in looking at how water use efficiency varies between the environments that these three species inhabit. So oak tend to inhabit low elevation, warm, hot and dry environments. Beech tend to inhabit more intermediate environments, and then the spruce inhabit high elevation, wet and cold environments. So by studying water use efficiency through these species, we can also look at how it varies in the environments that these species inhabit. So the objectives for our study were we sought to determine the spatial and temporal patterns of water use efficiency across the forests of Switzerland in order to provide new insights for decision makers. These decision makers range from government officials to the individuals that are actually involved in the forestry industry within Switzerland. Switzerland is covered by roughly 30% of forests, and the timber and lumber industry is really large there. So there's large financial implications as well. And so you may be wondering, I did use the International Space Station for this study, but I've never stepped foot in Switzerland before. I've actually never been to Europe. And so my bedroom over the course of this pandemic and my research project has basically become the Huntington Beach Center for Swiss Forest Studies. And so without EcoStress, there's no way this project would have been possible. And so I just want to give you some insight into how much EcoStress actually helped me with this project. So with collecting data with EcoStress, every time that that EcoStress passes over Switzerland, so roughly when it takes one image of Switzerland, it accumulates roughly 7,000 pixels of data with each pass. And EcoStress passes over Switzerland every 3 to 5 days. And we looked at 3 summers worth of data. So that comes out to roughly about 1.9 million data points. So these 1.9 million data points are the equivalent of 1.9 million on-site measurements. So if you were to do this project and try to replicate the data that I worked with on-site, and say we could visit 10 sites per day across Switzerland, a single map, so a single image that's what the EcoStress takes in one day, would take 2 years. And if you wanted to emulate the 1.9 million data points that I used for the 3 summers that we analyzed, it would take you roughly 520 years to get that same data set. So in conclusion, I basically got 520 years of manual labor. I put in the request, and in less than 24 hours, that data was available to me in my email. So that just emphasizes that EcoStress is such a novel and amazing new instrument as far as studying water use efficiency. And so a little bit more about the data that we worked with. This is just an image of me dropping a shapefile of Switzerland into the website. And then I just requested water use efficiency, and 24 hours I had all the data I needed. So again, we worked with the 2018 to 2020 summer growing seasons. And when you request water use efficiency data from Switzerland, it comes to you as gross primary productivity divided by evapotranspiration. We chose to recalculate that to just gross primary productivity divided by transpiration. And I'll go a little bit into more in depth about why we did that in the following slide. And then lastly, we compared these corrected water use efficiency values with the 182 sites that are around Switzerland. Those sites measure different environmental factor values like precipitation, elevation, temperature, etc. So we compared the water use efficiency that we got from the satellite to the on-site observations. And so just kind of talking about why we chose to convert water use efficiency to corrected water use efficiency. So it changes the mean estimate of water use efficiency, it increases, and it also leads to a more even distribution of values. And then the main reason why we ended up deciding to change to corrected water use efficiency is because it solely reflects the biological processes. So it gets rid of evaporation from the soil, which isn't necessarily a biological process that's occurring within the plant. And that's what we wanted to focus on. So by converting to just gross primary productivity divided by transpiration, that allows us to only focus on biological processes. And so this is really the culmination of what seems like 10 years, but really has only been a little less than 1 year of work and learning how to code in R. Dr. G and I have spent a lot of time producing this image. So this is the 1.9 million roughly data points kind of put together, and this is the median map of summer water use efficiency across the entire country of Switzerland for those 3 summer growing seasons that we looked at. And so just from this graph, you can kind of tell that there's a little bit of a pattern going on here. So as we go from a drier to wetter gradient from the north to the south, water use efficiency tends to decrease. Anywhere that's white on this image is non-forested regions. So the colored regions that we wanted to focus on are the forested regions. So this big chunk of white right here is actually the Alps, and the Alps are above the tree line. So that's why they're not colored in. There's no forest there. And so as we approach the Alps and elevation increases and temperatures decrease, we also see that water use efficiency decreases in this area. And then in the southernmost point of Switzerland, water use efficiency actually increases again, and we believe that's due to a dry Mediterranean environment. So these are the rough patterns of water use efficiency that we observe over Switzerland, but we also wanted to look in a little bit into how that varies between species. So this is a map of how water use efficiency, the seasonal patterns of water use efficiency for each 3 different dominant tree species that we looked at. So we have oak, beech, and spruce. And we can see that roughly with all the species that we looked at, there's a sinusoidal kind of pattern going on as summer begins and ends. What is noteworthy though is that the different species tend to have different variability in water use efficiency. So you can notice that spruce, which is the more purple color, has a much less distinct pattern than other species like oak. And we wanted to measure that in another graph, and we show that here. So this is just a box plot of those same water use efficiency values. And so we can tell that as oak, we go from oak to beech to spruce. And again, these 3 species inhabit their own environments. So it gives us insight into how water use efficiency varies at different environments. So you can see that oak has the highest variation as well as the highest median water use efficiency, and then down to beech and spruce. So as we move from deciduous species and then we move to evergreen species, water use efficiency tends to decrease as well as the variation of water use efficiency. And again, that also gives us insight into the environments that these species inhabit. And then lastly, we looked at water use efficiency and compared it to the different environmental factors. So elevation, slope, mean annual temperature, and mean annual precipitation. These measurements were the measurements taken on site in Switzerland. And then the water use efficiency on the y-axis is from the satellite data that we requested. And so these are 4 of roughly 9 environmental factors that we looked at, but I just wanted to focus on these 4 for this presentation. And so with elevation, slope, and mean annual precipitation, we see that there's a negative correlation. As those environmental factors increase, water use efficiency tends to decrease. And then with temperature, as temperature increases, we tend to see that water use efficiency tends to increase as well. And what was interesting that we found too is we also ran these linear regressions with aspect, nitrogen deposition, oxidized nitrogen, and reduced nitrogen. Nitrogen deposition has been increasing in Switzerland due to the use of artificial fertilizers over the past few decades. However, with our measurements, we were not able to find any evidence of a significant relationship between water use efficiency and nitrogen. And this is really surprising. And so that's definitely part of our next steps to kind of confirm and maybe investigate that relationship a little bit more. But that was definitely a surprising find. And so to conclude, for our temporal patterns that we saw in Switzerland, there are clear seasonal water use efficiency patterns. Spruce appear to have much less distinct seasonal patterns than oak and beech. For our spatial patterns, median water use efficiency and variation at sites increases at lower, warmer and drier climates, while decreasing at higher elevation, colder and wetter climates. Topography was a stronger predictor of water use efficiency than temperature. So that just means elevation and slope. And then there are clear differences in water use efficiency between deciduous and broadleaf species, which indicates that not all species will respond equally to climate change. So because we observed that oak species tend to have higher water use efficiency and a broader range of water use efficiency, we predict that as temperatures increase, those species will likely respond better to increasing temperatures than a species like spruce. And then lastly, climate predictions for Switzerland indicate temperatures will increase while not necessarily becoming wetter or drier. Due to our linear regressions, we believe that temperature alone may be enough to increase water use efficiency in the future. And so just to end, I would like to acknowledge that this work was funded by NASA and the Swiss Federal Office of the Environment. I'd like to give a huge shout out to Dr. Goldsmith for giving me this opportunity to work on this project during this crazy pandemic where all my plans for undergraduate went out the window in a span of what seemed like a week. So I really appreciate that. A huge shout out to the Biology 499 squad, Dr. Keller, Luke, and Kendra for really helping me figure out the story that I wanted to tell with this project and just really polishing off my project. I do want to give a shout out to Dr. Tamian, who's in the crowd as well. I spent my first 2 years of undergraduate working in his lab, and that's where I really got a taste for what research was, and he really helped me determine that's kind of what I want to pursue in my career. So without him, I wouldn't be doing this project or where I am today. So I do want to give a public shout out to him as well. If you guys have any questions, I would love to answer them, and thank you all for listening.
Thanks, Brandon. That was a great presentation and just non-trivial to go from working in Dr. Tamian's lab to such a very, very, very different topic. And anyone that has spent any significant time with remote sensing data and R knows that your launch of the Huntington Beach Center for Swiss Forest Studies was no small feat. We have time for questions for Brandon. If you're welcome to pull yourself off mute or drop something into the chat as you see fit. Dr. Tamian.
I have a quick question. First of all, I want to say that I think this project would benefit a lot if you actually do this in the International Space Station. So I would suggest that you request that in your next grant. So
definitely.
My next question is, you said that this result, this exciting result that you presented is going to help the governments and different agencies with their decision making. So can you give us like specific examples of how or what would those decisions be?
Yeah, definitely. So Switzerland is roughly 30% covered by forest, and timber and lumber production is one of their main industries in Switzerland. So maintaining the health of their forest from a financial standpoint, as well as just the natural health of the country, is really important to the government. So by aiding and providing this water use efficiency information and kind of letting them know what species might or might not respond to future climate model predictions will definitely be useful in their ability to adapt to future climate change.
Other questions from the crowd, or a follow-up, Dr. Tamian, as you see fit?
I mean, if I can ask another question. I was curious, how were you able to differentiate between the different species? Because you showed that figure looking at the water use efficiency in different species. Was that something you were able to do from the pictures, or that was provided for you?
Yeah, so that was provided. So across Switzerland, there's 182 on-site stations that take consistent measurements, and one of those measurements is what dominant tree species are prevalent in those on-site locations. So in addition to just taking like scans across Switzerland, we also requested just point data from those 182 sites. So when we got that point data, we would compare the water use efficiency values that we got from the satellite, and then we also knew what the dominant tree species was in that on-site location. So that's how we were able to determine that.
Dr. Scherf, I see your hand there.
Okay, so as you were going through, I was thinking about, you're talking about over the season, and I was wondering, and when you specifically mentioned that there was less seasonality with the spruce is an evergreen, so the needles are there all year round, whereas with the deciduous trees, right at the beginning of the season, there's going to be like no photosynthesis because you don't have any leaves on the trees, and then it's going to change. And so I was just wondering, is there some sort of, when you're looking at the different species, is there some sort of relationship to say like how big the final leaves are, or how many, can you look at a tree and kind of get an idea of its efficiency and the change by just what the leaves look like?
Yeah, that's a great question. We haven't thought about if the size of the leaf or what it looks like might impact the water use efficiency patterns. However, we do have like a rough kind of explanation for the overall seasonal patterns that we do observe. So, and I can kind of go into that for a little bit too. So I'll go back to, if I can, the seasonal map. So we believe that, and this isn't really literature backed or anything, so kind of why I didn't mention it in my project, but one of the possible explanations that we have for the overall kind of seasonal patterns is right when leaves are first growing in June, we believe that water use efficiency is high because they keep their stomata closed because they want to avoid kind of wilting out and drying out when they're first growing. And then it slowly decreases as the summer goes on and the stomata open. And then as temperatures rise again from July to August, stomata will close to avoid losing excess water due to transpiration. And so we believe that might explain the increase in water use efficiency. And then it dips down again as September goes on, and that might be due to just the leaves senescing at the end of the season. Again, we have a little bit of more work to do that. That might be kind of a combination of biotic and abiotic factors that might explain the seasonal patterns that we observe. But again, there's a little bit more work to be done with that.
Got it. Thanks. That's interesting.
Thank you.
I think we've got time for if there's one last brief question from the crowd. Dr. Keller, I think
I'll ask one, Brandon, just because I know you were really looking forward to hard questions as a part of this series. That was an excellent presentation, really, really well done. So let me start with that.
Thank you.
When you run all of your regressions with these environmental factors, it looks like you did that and you just sort of threw all the species in. And so some of those dots are oak and some of those dots are spruce and some of them are beech. Have you had the chance, or do you have enough data to look at these relationships within species? So, could there be a nitrogen deposition effect for the oak if nitrogen deposition is more important in kind of the lower areas of Switzerland? Have you had a chance to explore any of those relationships?
Yeah, that's a fantastic question, Dr. Keller. So that's actually, I have it written down right here on my notes. That's like the next steps that we're going to do after this presentation. So we do want to see if there's differences in relationships between water use efficiency specific for different species and the environmental factors that we measured, because like I explained with Dr. Tamian, we do have all of the water use efficiency point data tagged with what species it's correlated with. So we'll easily be able to run linear regressions with the different species. And hopefully this gets published. You can read about that in the final product.
Great, thank you.
Thank you.
Brandon, I join everyone in the chat in congratulating you on a great presentation, on everything that you have achieved in the last 4 years. I can't wait to see all the things that come, even if you forego remote sensing and move back towards molecular genetics. I will still be friends with you and Dr. Tamian. Let me briefly say thanks to, as I did before, to Dr. Fudge and Dean Lyon and to Dr. Carter Berry, who is hiding in the background here, for all that you've done to mentor students in this last most unusual of years. I am so sort of floored by all these presentations and everything that was achieved in such unusual circumstances. And even if we were all sitting in the same room, I would still say that to see students that have wrapped their heads around both, you know, microgels and advanced microscopy, remote sensing and plant physiology, building greenhouses, you know, in their backyards, and learning about the physics of light is really quite impressive. And I hope that these were really fantastic learning experiences for you, and that you know that we will look forward to celebrating all of your successes for many, many, many years to come.